One Health Around the World series. My name is Dr. Steve Osofsky. I'm director of the Cornell K Center for Wildlife Health, and with our partners at Cornell University and the College of Veterinary Medicine and our great partners here at eCornell, we're really glad you've joined us today. This is actually the third episode in our series. As a reminder, our first webinar was about the work of our colleagues at the Cornell Wildlife Health Lab in North America. Then we moved on to Asia and focused on examples of our efforts in Nepal. Today, we're going to pivot down south to the Antarctic and Sub-Antarctic regions. But before we do that, I want to make sure you all are aware of what the Cornell K Center for Wildlife Health is all about. Our vision really is a healthy future for wildlife, people, and planet. And our mission is about transforming science into impact through discovery, education, engagement, and policy to ensure a healthy future for wildlife and the environment that supports us all. Why do we do these things? Because we need nature and nature needs us. Now, what do I mean by that? We need nature for all of the incredibly important ecosystem goods and services nature provides for us, from clean air to clean water, to climate stabilization services, to pollination, things that make our food systems sustainable, that humanity ultimately depends upon. And when I say nature needs us, I often get pushback. Oh, nature doesn't need us. Nature will be just fine. But I disagree. Nature needs us to be better stewards, to be more mindful. We can't mine natural resources. We need to live off the interest that natural capital generates so that future generations can continue to benefit from ecosystems and all the biodiversity that humanity ultimately depends upon. I want to quickly now summarize the 4 pillars that capture much of the work that the team involved in the center is involved in. Most of us do work that responds to the health threats to the survival of key species. Many of us work on providing landscape-level guidance on wildlife-compatible land use, policy, and practice. That might be at the state level, national level, or regional level in partnership with a multilateral organization. A lot of our work is about minimizing disease transfer at the interface between wildlife, livestock, and ourselves. And this is a good segue to make sure that everyone understands the definition of the term One Health. One Health is really, for me, it's an approach. It's about recognizing that our health, the health of our wildlife, the health of our domestic animals, are all inextricably linked and also underpinned by our approach to environmental stewardship. This more holistic, multifaceted way of thinking about these complex problems really gives us more entry points to try and solve those problems. All 3 of those pillars really roll up into the last pillar, which I call building new constituencies for wildlife conservation. And what I mean by that is in many parts of the world where we work, governments may be strapped to even provide the most basic services for their citizens. And in those situations, conservation can be a luxury, and I totally understand that. But part of our role is to use good science to show how biodiversity conservation can improve public health outcomes, can improve options for agriculture and sustainability in food systems. So if we can make conservation more relevant to these other sectors, we can start to engage not just ministries of environment or departments of wildlife, but departments of agriculture, ministries of public health, and even ministries of finance. I now want to introduce, we have 2 great speakers here today, and we're going to now focus on the Antarctic and Sub-Antarctic regions. And our first presenter is Dr. Amandine Gamel, a disease ecologist and veterinarian who's been working on One Health challenges throughout her career, focusing a lot on diseases threatening remote seabird colonies in the Southern Ocean. Now, Amandine, we were talking before we went live. I apologize that when you got back to Ithaca, it was colder here than in Antarctica. So I'm sorry. I'm really glad you're here, and I'm really glad you're a member of the team at the Cornell K Center for Wildlife Health. We're going to give you about 25 minutes to update us on your work on avian influenza and some of the other threats facing marine birds and mammals in the Antarctic and Sub-Antarctic regions. So over to you, Amandine.
Thank you, Steve, for the introduction. Thank you, everyone, for the opportunity to be here. And so, Steve was talking about One Health, people and animal health being connected. It might not be obvious how wildlife that is so far away from us, all the way down to Antarctica or Sub-Antarctica, can be connected to our health. So this is a paradigm that Steve talked about. We have human health, animal health, and environmental health all together. Most of us are living, out of Antarctica, the rest of the continents. Very few people are living in Antarctica, mostly on scientific stations. So how is it all connected? And why should we care about health of the Antarctic ecosystem? So usually we don't need much of an explanation. There are lots of penguins there, and most people love penguins. They are icons of pop culture, flagship of Antarctica. They're cute, they're fun. Yeah, most of us love them. And there are a lot of penguins there. So this is, for instance, a picture of a colony of king penguins, and you can see it's thousands of individuals that are packed together. And so we have lots of big, very dense colonies of seabirds like that in the Antarctic and Sub-Antarctic region. And we also have other species that we could care about, so seals and other seabirds. Obviously, there are also in the Sub-Antarctic region some plants and insects. And there's one specific species I want to talk about now, because when we talk about One Health and the impact of humans on the planet, we often think about climate and carbon. And Antarctic krill might be, understudied, like getting very little interest, but it's an important factor to consider in the health of our planet. So what Antarctic krill is exactly doing is capturing carbon that is dissolved in the ocean, so coming from the atmosphere, dissolved in the ocean, and then being captured in first phytoplankton and then krill, and then it's captured in biological material that will stay at the bottom of the ocean. And so, we talk about the Amazon being the lung of the planet, but we also have krill being that hold of carbon for the planet. And it is a lot of carbon. So if, like me, you're having trouble realizing what this means, maybe an amount of dollars would be more helpful to understand. So it's been estimated that the ecosystem services provided by Antarctic krill are about $15 billion per year. So again, that's involved in helping mitigating the increase of carbon in the atmosphere, and so ultimately helping us having a healthier atmosphere and mitigating climate change. And so how is it looping back to penguins? The phytoplankton that is first capturing the carbon that will end up in the krill and then at the bottom of the ocean is actually also using lots of resources from bird guano that can act as fertilizer of the ocean. And closing the loop, birds are also affected by the climate on which our activities are having an impact. So to give you an example, here are some penguins on which some movement trackers were put to see where they go find their food. And scientists have noticed that over the years, as the concentration of sea ice and the location of sea ice in the Antarctic is moving, penguins might have to go further away to find food because they find most of their food, which is krill and plankton, around the sea ice. And so as they have to go further away to find food, it makes just their life easier. It makes their life harder. They have less energy and they can invest less in reproduction. And this goes in parallel with a decline in the population, notably of this specific penguin species, the Adelie penguin. So, it's a bit grim. We are seeing climate change impacting species that are as far away as penguins in Antarctica, having a bad effect on, both them and the other organisms they feed on, so the krill and the plankton. But the good news is that because there are all those complex interactions that can, lead to bad effects, it also means that if we manage to address them, usually we address several problems at the same time. So we can have win-win solutions to both preserve penguins and krill at the same time, leading to then a healthier atmosphere, notably through carbon sequestration via krill. But in the meantime, because we're not there yet, some of the penguin population are declining. And the thing with smaller population, besides the direct impact that we are measuring as it's happening, it's also that it makes them more vulnerable to further threats because obviously they're not just facing one threat at a time. And so, for instance, on top of having to go further away to find food, we can have warming events that create extra heat stress for them or change the conditions in which they're breeding. And it's taking me to one of the threats that we all have in mind at the moment in the bird community, and that's infectious disease, and in particular, avian influenza. So you can imagine that when you have colonies like that with all those packed birds, if a pathogen makes its way through the colony, it can very quickly affect a lot of animals, spread really fast, and kill a lot of them. And it's a case for most seabirds, so most colonial seabirds. So I was showing you penguins, but here is the same with albatrosses on a Sub-Antarctic island, which also form very dense colonies. And so some diseases have always been there. Diseases are, they're part, like parasites are part of biodiversity the same way birds are. And so, for instance, in Sub-Antarctica, we have known about pox viruses that are affecting the birds. Also, ticks are present in the environment and can go on the birds and transmit pathogens to them. And we think from some recent studies that those kind of pathogens have always been there, or at least for a very long time. But some threats are more recent, and I was talking about avian influenza. So at the moment, we're experiencing the biggest global outbreak of highly pathogenic avian influenza. And it's big in terms of places that is reached. So it originally emerged in Southeast Asia, in poultry farms. So that's why I'm saying it has that connection with human activities, because it's not a virus that emerged in wildlife, but really a virus that emerged in industrial farming. And then spread through the world with a mix of transmission to wildlife and transmission to poultry, with some evolution in the meantime that made it more transmissible. And so it reached further places that it had ever reached, but also more species, and including a lot of seabirds and also marine mammals. And so in 2023, so a year and a half ago, we saw the virus making its way down through South America. And there was that threat that it would eventually reach Antarctica, which it did. And I will focus on a location that is just between South America and Antarctica, and that's the Falkland Islands, or Malvinas, where I've been conducting some research on bird health over the last few years. And so we could track in real time the introduction and spread of the virus there, because we've been working there for a few years, as I was saying, and we were expecting the virus to come. So we started collecting data before it even reached the islands. And what we could see in October 2023 was first one outbreak, and then another one, and another one, and another one. And so ultimately we had several sporadic cases, so isolated cases here and there, and 3 big outbreaks that affected notably gentoo penguins and black-browed albatrosses, and caused the death of tens of thousands of those birds. And so what it looks like when you have an outbreak, it's something like that. So here you can see an albatross and penguin mixed colony, and you can see lots of empty nests, and all those empty nests are birds that died during the outbreak, and on some of the nests you can still see the carcasses. And so you can see that it's very localized, but in that little sphere, like that little area, it like all of the nests are empty. So it shows really the intensity at which it was spreading locally and affecting the birds. And we went back this year, and we can still see the effect of the outbreak. So for instance, we can see now some penguin colonies that are rebuilding themselves, but you can still see the carcasses from the penguins from last year. And so the questions we're trying to address is, first, what is the real extent of the outbreak? Because obviously we can see the birds, we can see some dead birds, and we can see some empty nests, but we don't know if some more birds are affected and actually surviving, like when we have, just a seasonal flu. And also, if there are some birds that are surviving, can they be involved in the spread of the virus? So what we're doing is that instead of only focusing on the dead animals, we're also looking at the live ones that are around. And so we focus on 2 specific group of species in the systems. On one side, we have the penguins and albatrosses that we call mesopredators, because they're mostly, so they prey on fish, so they go at sea to find their food. And on the other side, we have the apex predators, so gulls, skuas, and bird of prey like caracaras that will feed on the other birds. And we separate them this way because we think that if you're an apex predator, and a lot of them also scavenge, because you're feeding on other birds, including dead birds, you're more likely to be exposed to the virus. So maybe they've evolved something to tolerate this kind of infection, or just maybe they're more dying because they're being more exposed. So then we go in the field, we collect samples, so we grab the birds. Sometimes the birds grab stuff from us. So you can see here a curious caracara that was monitoring us while we were doing sampling. And so in that little box we have in the corner, we have samples that are on one side, blood samples that we take from the birds to see if they have antibodies against the virus. And if they do, it will tell us that they got exposed to the virus and survived. And we also collect swabs so we can look for the virus itself, kind of like we would do when we do ourselves a COVID test. And so I'm going to show you some of the data we're getting. So I'm going to be showing maps, and I want to point that when we have a blue point, it means the bird that we sample here didn't have any antibody against the flu, so probably had never met a flu virus before. And if it's a red point, it means that the bird had antibodies against H5 in particular, which is the subtype of highly pathogenic avian influenza that is currently spreading across the world. And so we can see that before the first outbreaks in the Falklands, we had happy, healthy birds that had never met avian influenza before. So we have all those blue points for the different species, albatross, penguins, and scavengers. And then when we came back a year after, we had mortality events that were affecting the albatross and penguins, that's the picture I showed you before. But despite that, none of the birds had antibodies. And so what it tells us is that we knew the birds were dying. We confirmed that they were infected with the virus, but they were not getting any antibodies. So probably it means they're dying before they get a chance to mount an immune response. On the other side, we have the scavengers that were not dying so much, but they all had antibodies after the outbreak started. So this is telling us that they are heavily exposed, very likely by scavenging on the dead penguins and albatrosses, but then they survive the infection and develop some immunity. So then from there, we try to look at, okay, we found all those scavengers that were tolerating the infection and telling us that they were, you could still be alive after having encountered the virus. So actually how widespread the virus was, if, monitoring the dead birds is not enough. So we sampled some other birds on other islands, and you can see here all the silhouettes that I added, the yellowish silhouettes. That's bird species on islands where we had not detected any cases. So in some islands where we had no outbreaks, we still found some birds that had antibodies against the virus. And also for many species that were not dying from the virus, we found some antibodies. So it's telling us that the virus was way more widespread, and again, that monitoring only dead birds is not enough. We need more systematic monitoring, or also the live one, if we want to understand the dynamic of the virus there. And so if some of those birds are surviving, then it also asks the question of what's going to happen in the future, is the virus going to stay around, and is the virus going to keep on spreading? So to summarize what we are seeing between the different species, on one side we have the penguins and albatrosses that are not developing immunity and are heavily dying from the infection. And on the other side, we have the scavenger apex predators in general that are being exposed to the virus but seem to survive and not die, and that we can use then as sentinels to track where the virus has been circulating. So now, talking about where the virus has been circulating, how does it spread? So here you have a very complicated tree that you don't have to focus on the details, but the idea here is that every line is a virus that we got from the field, and we built with that a family tree of the virus by doing some genetic sequencing. And what we could see from there is that within the Falklands, we had just on one island, so that island called Steeple-Jason that had the biggest outbreak in the albatross colony, we had offspring of the virus that were related with some other offspring that were from South America. So I mean there were several introductions of the virus coming from South America. So just at the scale of that one tiny island, the virus managed to come several times, which highlight the fact that there is a strong connectivity between South America and this island, which is at the crossroad to Antarctica. And we also had a second outbreak in the southeast on Sea Lion Island, which we could link to one of the outbreaks from Steeple-Jason. So this tells us that once the virus was there, it could then further spread across the Falkland Islands. And it's also telling us, because all those virus samples that we got were coming from different species, that there is a lot of cross-species transmission happening. So notably between mesopredator species that are sharing a colony, so for instance penguins and albatrosses, but also with scavengers that are feeding on those birds, and then potentially spreading the virus to other species that are around. And so how did the virus manage to come that often to these islands? To try to get some elements of answer to that question, we gathered some data from previous studies that looked at how animals are moving in the region. And you can see here 3 species: albatrosses, fur seals, and penguins. So black-billed albatrosses, Sub-Antarctic fur seals, and Magellanic penguins that are moving very regularly between the Falkland Islands and South America, just as part of their normal searching for food strategy. So it's just when they have a nest on the colony, for instance, for the albatrosses that is on the Falkland Islands, but then they would do those back and forth toward the coast of South America because that's where they find the best food for them at this time. And they can do that trip in just a few days. So even if they die quickly after being infected, they could potentially have the time to go there healthy, get infected, and then fly back, and on their way back bring the virus. And so the following question was, now that the virus has been introduced several times and, settled in those colonies in 2023, can we expect it to stay around in the system? And so we did the same exercise of building that family tree for the virus. And we could do it this year very quickly because we got some support to deploy some field laboratories. So we could really sequence the virus in the Falkland Islands without having to bring back the samples to the US. And we could see that the offspring of the virus from this year are actually related to the outbreak that we found last year. And another interesting point is that it's the same branch of the family tree that we find in the penguins and now in a new species that we find infected in the Falkland Islands, which is elephant seals. And so this is telling us 2 things. The first thing is that it looks like the virus stayed in the system. Otherwise, we would have expected that, branch we built with the samples from this year to be connected closer to South America than to the samples from last year from the Falklands. But it is connected to the samples from last year from the Falklands. So it very likely stayed in the system. And we think this is probably through some species that are spending winter on the islands. So most of the colonial species in the systems are migratory, so they go spend winter at sea. And so you don't see them on the islands. But there are a few species, notably gentoo penguins, that stick around and could still spend some time on land, which could create enough opportunity for the virus to be maintained in the system. And then they also, share the islands with other species, including elephant seals. And so that's a spillover that happened. So like the transmission of the virus that we think happened from penguins to seals there. And, it brings us to the constant worry we have about the virus being able to evolve and to infect mammalian hosts like we're experiencing also in the US at the moment with dairy cattle. So, if we summarize, we have this complex network where we have different species, but the key message is that every species is playing a different role. So some species might be more involved in the introduction of the virus, some might be more involved in the local spread of the virus, and some in the overwinter maintenance. And we're not there yet, but it means that if one day we have a vaccine we could deploy, we know that those are the key species we should target. So for instance, if we want to have lower outbreaks in the year, we know that we should interrupt overwinter maintenance, so vaccinating gentoo penguins could be a solution. So here, I'm getting a bit ahead of what we're capable of, but the idea is by understanding those interactions and also by looking not just at the birds that are dying, but actually the other species that could be involved in the epidemiological network, we can come to thinking about mitigation strategies that could be efficient. And so if we go back to our original statement that Antarctic health is connected to the rest of the world, I was mentioning the krill and the atmosphere, and now we're adding a new arrow with disease that are potentially, so some of them are in Antarctica and might move around, but also some of them are emerging from notably farming or other anthropogenic activities and could make all their way down to Antarctica and potentially evolve and then spread back and infect, again, livestock species. So it can go in every direction, and it's all connected because, again, the health of the birds is critical in that network of interactions that are happening between where we live and Antarctica. And so that's my conclusion for this part. And so now I will hand out the mic to Steve.
Yeah, that was a lot of food for thought, Amandine. I'm going to ask you one question before we move on to our second presentation. We talked about One Health being a recognition that our own health, the health of wildlife, the health of domestic animals, the health of the environment, all inextricably linked. And I'm just wondering, in your experience, do you think we've gotten where we need to go in terms of dealing with the poultry sector? This virus, it's like a fire, and we have these massive, duck holdings, other types of poultry in Asia, other parts of the world, and they're just a great source for these viruses. And we're not even able to vaccinate them yet in many ways. There's vaccines, but there's all kinds of politics around vaccination. How are we doing in using the One Health tools to really address this problem? Because we're a long way from having the ability to vaccinate penguins.
So, I think there's politics involved, but also the logistics. Because when you think about the number of birds, like I learned that the average poultry farm site in the US is between 1 and 2 million individuals. So, it's also a lot of birds to vaccinate. So we're not there yet. And I think, even if we're thinking more and more in terms of One Health, there are several barriers that still exist. So the first is that ultimately the way we are currently managing wildlife conservation, environmental protection, human health, animal health is very structured, and there are a few links between. So for instance, in the US we have the NIH and the USDA and the Fish and Wildlife, and that's different institutions. They have path rails, but it's still... it's not made to work as one system. And then obviously this idea that we can do anything we want in Antarctica. If the threat is still coming from the outside, then it's kind of like wasted efforts, and we can do what we want in America. If there are some threats coming from Europe and Eurasia, then it's the same. So it's ideally we should all work together to try to address this. But I think as we are learning that One Health is more than just people bringing disease to wildlife or wildlife disease bringing disease to people. So when we talk about avian influenza in Antarctica being an anthropogenic threat, it's not like people bringing the virus. It's like something that started way before that with farming and so on. Yeah.
No, One Health really is about building bridges between disciplines and at the same time breaking down institutional or departmental silos, agency silos, et cetera. Well said. So we're going to move to our second speaker today. Marie Bouillou is a member of your team, and she is also a disease ecologist. Marie, you're currently looking at the role of introduced rodents on wildlife pathogen dynamics in some of the Sub-Antarctic islands. And as I understand your work, you're trying to assess whether better control of what are these essentially alien invasive species, these introduced rodents, could help lessen the impacts of some of these important wildlife diseases on the wildlife itself, but also potentially in terms of public health. So I'm going to hand over to you for the next 20 minutes, and it's a different take on another set of One Health issues. So thanks for joining us.
Thank you. It's really a good opportunity to talk about one part of my postdoc research. And notably, today I will present you how invasive rodents in Sub-Antarctic island is a good case study in the One Health approach, and we will discuss the challenge on its application. And scientists and the organization are raising alarm about the loss of diversity, and notably in the island ecosystem. And island conservation report 95% of bird extinction have occurred on island. And to understand that, it's important to understand in the island ecosystem is more vulnerable because we have a low diversity, we have a species specialized and a small population, and leading to a low resilience to different anthropogenic effects. And this observation is even more important in Antarctic region. We can observe the resilience is even more resilience is even more important because we have environmental because the extreme temperature, for example, and isolation geographical is really important too. But one of the main causes of the decline is invasive rodent, and notably rats and mice. They have introduced by human and notably by maritime transport, and continued their migration on the island with the really high capacity to swimming. And because we have ecosystem island is very vulnerable, and we have low density and diversity, and we have not many predator. And the second step is the establishment of rodent. It's they could be survive in the winter in extreme temperature, and there could be to do with the impressive trait of life. For example, they could be building management or of burrow or other trait of life, for like a various jet or like various morphology. And they have a very high level of density and a high level of reproduction. And for that, they can have a really good adaptation and different change, and for like anthropization disturbance, for example. But numerous studies show the rodent have a really big impact of seabird, and notably it's observed everywhere in the world, and every island, and on the Antarctic region is more important. For example, in Gore Island and Marion Island, we can observe the really important level of predation, the predation for the eggs, the predation for the juvenile or adult albatross or giant petrel. And other studies show the indirect impact by disturbing their habitat and resource. But another way to explain the bird decline is a new infectious disease. And we can observe a new infectious disease in the world, but in the Antarctic region too, with highlight pathogenic avian influenza, tuberculosis, salmonellosis, and avian ehrlichios or avian botulism, avian cholera. And this disease increase in the time, and it's also true in Sub-Antarctic region, for example, in Falkland Island, where the Amandine Gamble's team have observed a poxvirus outbreak in 2024. But we can ask if the link between both observation, if, for example, if invasive rodents play a role in the degradation of animal health, and notably in the infectious disease for the bird, in addition to their disturbing effect on the ecosystem. But we know the rodents pose a high zoonotic risk. The zoonotic risk means is when we observe a transmission between the animal to human, and we can consider the rodent like a important reservoir species due to the fast alive trait of life. They are the high density and high reproduction, but they have not a human system important. It's really weak, and for that they accumulate the different microbiome and a pathogen, and without a symptom. And secondly, there are the really a diversity and habitat and the contact, and for that it could be considered like a bridge between different wildlife and the human, or for the domestic animal and human too. And for that it's considered like a super spreader. But this is really anthropocentric perspective. And in the literature, we have the important link, because it's not only the transmission between animal and human, and generally we can observe interspecies transmission between different species, and it's possible the rodent have not only the impact on environmental health and human health, but could be have maybe impact on animal health. And we can form a new hypothesis. We can suppose the rodent have carry a different pathogen for avian, and we can suppose the rodent could be a good vector for different population of bird. And in the time or in different colony, for example, or the bridge between different species, therefore different species of bird, but with other animal in the wildlife. And this process, it could be explained by different method of transmission, like with by intermediate of environmental, like the water for leptospirosis, or other pathogen could be transmitted just because we have the same habitat, and we can observe just a contact or aerosol from the rodent. And other process is mediated by trophic interaction, notably by predation, and we can suppose this transmission could be explained, the transmission of toxoplasmosis on salmonellosis or avian cholera. But the World Health Organization recognizes the interconnectedness of human, animal, and environmental health. And it's for that, it's not only a transmission process, but because we have the impact for environmental health, we can have for that increase disturbance of stress for animal, and the change in interaction between different species in community, or in change in diet or habitat or microbiome, for example. And for that, the animal is more susceptibility. It's maybe a new hypothesis to explain the transmission or the susceptibility of bird with avian influenza. But we can see it's really important to adopt a One Health approach to better understand the dynamic of pathogen circulation among animals and wildlife. But if rodent impact One Health, then managing the population could provide a sustainable and holistic solution, notably for One Health. But to allow better protection, we need to understand the mechanism, and we need a better surveillance. But for this, it's a big challenge, notably to monitor the ecosystem health. We need a local collaboration with many organizations and many stakeholders, citizens, and it's the case in the Falkland Islands, where we need many collaboration. And for that, we understand the diversity and the health of different albatross colony or other bird colony. For the other challenge, it's when we need a better surveillance for invasive species, and notably for the rodent. But for the rodent are everywhere. We have the trace, we have the burrow, and we have many information there, the presence. But it's not easy to trap, and it's a problem. It's like really intelligent species, and sometimes they hide, or just because the density varying in the time and different habitat. And generally, for the surveillance of animal health, it's not easy because the sampling is really extreme in different extreme conditions, and we need expert for trapping a bird or rodent. And the field laboratory is not really in the big laboratory. We need to use a field laboratory, and after that, we need to bring your sample in the big laboratory in Cornell University, for example, for bird safety level 2 detection of pathogen. And we need a different methodology for detect a large kingdom of pathogen for bacteria, parasite, virus, and you need a sequencing methodology or a serological methodology. And for that, we need very long time to know the health, animal health surveillance, and generally it's for that we have the information just after the outbreak. It's more like a reactive process, and the research is more for one pathogen and one species after this outbreak. But we need today have a proactive surveillance, and notably it's like a fire. If you prevent the fire, if we prevent the outbreak, we can contain in the small population or small fire. But it's not easy if you have a pandemic, and it's for that we need improve the surveillance in the local surveillance and very proactive surveillance. And for that, we have a new method, and it's one part of my project. It's to try with environmental DNA to detect the invasive species in the water, for example, and pathogen. And we can observe the pathogen too in the feces, for example. And we can have and the objective is to apply this method for local organization and institute. And maybe it could be possible if you couple this method with minion or nanopore sequencing, because it's really portable and it's more easy to use. And it's for that, it's a new method for more sustainable and proactive surveillance. But there are the last challenge. If you have a better surveillance and we understand the dynamic of pathogen, it's not easy too for the management of population. But the good news is if there are the success in the management, we have a really good success in long-term restoration of avian communities on the highland ecosystem. But the rodent control method often fail. And for example, in the Falkland Island, we can observe in the map, we have just a little small success in for eradication, but not for the big highland or continental highland. And that explained because we need many resources for the success of this method of eradication. And the cost and logistics is really important, but we can have a different priority for the citizen. And it's for that we need to take into account the societal aspect that influence the management and the local policies. But we have a big, we can observe a balance between the benefits and the cost. And the another cost exists. It's when we use the chemical methodology for eradication. There could have a cost for other animal because for example, SQR or scavenger could be hit the rodent and they have a danger for the health. And we need to take into account for that the indirect effect for all species. And just after this, we need to take into account ethical aspects. That means we need to take into account a new perspective, because if you continue of the habit, the human habit, you have a possibility to continue the invasion of invasive species. In this due to habit human, there are the many invasion of rodent and other invasive species, and we have the vulnerability to ecosystem, and notably the highland ecosystem and the bird. And for that, it's more easy for a rodent for establishment and invasive the highland ecosystem. And so invasive rodent is really a good model for understanding the role of invasive species in the One Health in general. And we need for an integrative and sustainable approach to protect health. And acting against invasive species means protecting not only ecosystem, but for the impact on the and acting for the human and the animal health. Thank you.
Great. Wow. Let me. That was a lot. So it obviously, for those of us immersed in conservation, dealing with rodents in particular on island ecosystems is not a new problem. But what you're bringing to this is not only is it their direct impacts in terms of egg predation and taking young birds, but also now the various ways that they could be spreading pathogens. And it sounds like, from a One Health point of view, that you're making the case that if you can show that some of these same diseases are also of concern to people who might be living on those islands, you might be able to pull what I would describe as a bigger lever arm to influence policy decisions. Is that a fair takeaway from your talk?
It's just an hypothesis, but I think, yes, it's really important to discover this if in rodent impact not only ecosystem but the animal health with infectious disease. Yes, and for that, yes, if we have these both effects. If you manage the rodent, we can have a better animal health and ecosystem health, and it's a really sustainable solution.
Yeah. Well, we have quite a diverse audience, so I wanted to maybe get a little bit more detail on the tool that you described, the use of environmental DNA detection. What can you learn from environmental DNA? How long can a species have been in a body of water and then flown away, and then you can still find it? Or in the old days, you could only detect pathogens by sampling either directly on the environment or the animals themselves. But it seems like eDNA, particularly with water and now air, is opening up whole new possibilities. Can you say more about that?
Yeah, it's a good question because just now we have a method. It's more like we have a dissection, and after that we bring the samples for different organ in the laboratory, and from different organ you need to detect different pathogen. For example, the kidney, we have the leptospirosis and the other, but it's a really long time for that, and maybe we can observe a pathogen in the water and the surfaces, and it's an hypothesis too. I don't know. It's a really a good methodology, but I hope because if it's the case, we can use this method. It's very more simple because. Yes, it's possible to detect a whole pathogen, a whole kingdom, and maybe the other and the diversity of microbiome in general.
Yeah.
And but it's not really easy to take in place this methodology.
Yeah. So we have. I have a million questions for you, but I'm going to. Be mindful of questions coming in from our live audience. So I'm going to throw a few questions here. We can talk about them, and then I might throw in some additional ones. Gina asks, is it possible that, and this would probably be for, well, either of you can jump in on any of this, but is it possible that the virus, and I think they're talking about high-path avian influenza, is able to survive in soil or freshwater between seasons rather than just be harbored by, say, a penguin? Given the cold months and things like that, what are the epidemiological characteristics of the virus in terms of environmental survival?
So it's a question that comes often because obviously when you see lots of carcasses, you're thinking, okay, it could maybe stay in the carcasses, stay in the soil. It's been like so far we haven't found any evidence. I think the longest where people have tried like after an outbreak to sample was 2 months in Scotland when they had a big outbreak in seabirds, and they couldn't find the virus anywhere. But sometimes, as Mary was saying, like just within one rodent, sometimes a pathogen can be in the kidney and not in the brain. So it's the same with the environment. Sometimes if you're just targeting the wrong body of water, you could miss it. So we probably need wider screening until we can figure out if that's the case. Obviously, in places like Antarctica and Sub-Antarctica, but like Antarctica in particular, you could imagine that the virus could stay frozen in the carcasses and so reemerge when the carcasses are melting again. We haven't, I would say, looked too much for it yet. So we need to do more work on that. But there is also, other considerations. So it's colder, it's true, but also the water is salty, it's very windy. So there is also those elements that makes it very hard to predict how long the virus could actually stay in the environment. If it's in a carcass, like for instance, we know it goes a lot in the brain of the animal. So if it stays in the skull, it could be protected from UVs, but if it's outside, then not. So it's an option, but considering the species that were affected, because I said we think the Gentoo penguins might be the ones who kept it over winter. We also had some die-offs of Gentoo penguins over winter, so it's like several elements together. Like it's not just the one sequencing from the virus. Yeah, so possible, and then to see like what is the weight of each option. Yeah,
we've got to look at every possibility. So these questions cover a lot of different topics, and this is an unfair question because you guys are both focused on infectious diseases, but fair enough. Carol wants to know if you are seeing anything related to plastics or microplastics in the species that you're working on in this region.
So in the Falkland Islands, we are very spoiled that there is not too much of it. I think it's just a matter of the oceanic current. Like there is not too much plastic that is arriving there. So when we do find dead birds and we necropsy them, it's very rare to find plastic. But I know in some other places, they do, when they open chicks, for instance, they find that they've been fed literally with plastic. So with that said, I've never done a study specifically on that. And I don't think there has been any study in that archipelago specifically for microplastic, for which you will need to go something more than just opening and looking. So there might be more than what we are seeing, but it's definitely an issue with like different levels. Like I know, for instance, Midway Island in the Pacific, it's really big. So it depends on the lots of conditions, but including the oceanic currents.
Yeah. You spoke about multiple threats and then the hypotheses around rodents, and now we're talking about microplastics. So I guess this is kind of a personal question. Most of us get involved in this because not only are we interested in the science, but we're interested in making things better. Marie, you're a young scientist. What keeps you going? What gives you hope given the threats that you're seeing to these sensitive systems?
I don't know. I think I don't know.
Well, I guess what I'm asking is, you have a hypothesis, but I'm sensing, having talked to you at other points in time, that there is an opportunity for improving uptake of what you're learning, again, by involving the communities, the societal perspective, thinking about, you were talking about people don't like the idea of using rodenticides to get rid of rodents, but that might be a solution as long as you're mindful of potential environmental impacts, impacts on people, their pets, their domestic animals. So I guess I think one of the perils of our webinar series is it can sound very gloom and doom. And we're going to talk a little bit about pandemics because we can't avoid that in this discussion. But I guess if your hypotheses are true, then presumably you see ways to bring science into policy for positive change.
Yeah, it's really important for the perspective of social aspect and other thing. And better, yes, we need to learn of community and communicate the result and to learn from training, for example, or just to take into account maybe the conflict of interest, because it's important to understand why why this method is really benefits for One Health, but why it's not really used for everywhere or every island, because we have other social problem, and for that we need to take into account this problem to do the method of eradication, for example, or other method of control together with the citizen. And it's important because if we don't take into account this aspect, maybe I think maybe the method is fail in the time.
Yeah. No, I think you make a really important point. I think gone are the days where scientists could just publish their papers and think that if we publish it, people will use it. That's not the way the world works. And your work is an example of how important it is to integrate local perspectives, societal perspectives, ethical concerns, so that as you develop your science, the communities that are most impacted are part and parcel of the process, so that at the end, they understand what questions were asked and how they were answered, and they're more likely to utilize the information you generate. So I think that's a really important piece of advice for anyone who wants to do this kind of work, because almost everywhere we work, it's that interface with people in the communities in the places we care about. I want to pivot to a question from Jackie, who wants to know how likely, in your opinion, is high-path avian influenza to eventually become a pandemic. I think it's fair to describe it as a panzootic. The number of species that this virus is hitting now, from birds to mammals, I'm frightened as a conservation scientist and as a veterinarian. But from where you sit, what do you think?
I think this is a kind of question that we know we will always have, but we're always scared of having. Like, we don't like to make predictions because, what are the odds that we actually right? But no, it's like obviously a question that everyone has in mind. We see a lot of cross-species transmission. So I was mentioning from birds to seals, and it has happened also in South America. There were big outbreaks in sea lions and then in elephant seals. And there are other outbreaks affecting also seals in North America and in Europe. So it's definitely something that could happen. I think, considering the current situation, it looks like the threat is now coming mostly from what's happening in the US, I would say. And I'm saying the US because that's where we have the data. Maybe it's happening somewhere else too. But now that we have the virus that has, made its way to the dairy industry, which is an industry where there is a lot of contact between workers and the animals that are being infected. And that also because it's been spreading silently in the dairy farms, that's I would have given an answer 2 years ago, but now I'm like, oh yes, it's more likely now. And also, I mean, I remember when I was a kid that we were talking about avian influenza becoming the next pandemic. And now we're getting close. If it's not happening now, it's going to happen eventually. It's a virus that evolves very quickly. And, COVID came before avian influenza. But I think in any case, and that joined the point that Mary was making of we need better proactive surveillance, so we're not taken by surprise we've been taken by surprise by COVID, and now we're again taken by surprise by bird flu, even though we saw it coming. It's been spreading through wildlife for several decades, but really blowing up over the last 3 years, and now we're still surprised. We're having cases in humans, are we starting to having some? So yeah, I think in any case we should be ready for it.
Yeah, no, I agree with you. I feel frustrated in many ways that we didn't learn the lessons I was hoping we were going to learn from COVID-19.
But, you said things were a bit, gray, but, or black, dark.
Right, but, I want to ask you why you're hopeful.
Yeah, so, Mary mentioned it, and I also mentioned it, the thing is because everything is connected, the good thing is that if we find one solution to one problem, usually we tackle several at the same time. So, if I don't know what is the solution for that, but they're acting on livestock management. So for instance, we know acting on bird movement, that's really wild bird movement, that's really hard. We cannot just keep all the birds on one island. But things on which we do have control, like overfisheries, overfishing, livestock management in the US.
We're not powerless.
Yeah, these things. And then if we tackle those issues there, then it can have lots of cascading positive consequences.
So that's what keeps
Let's
me going.
focus on that.
Yeah. There are things we can do that we're
Yeah.
not doing, and we can do better. And I think we can beat these things back, but it requires that One Health approach, that bridging across disciplines, sharing information, and collaborating. I think we're about at time. And so I want to thank everyone who tuned in, and we're also going to record this webinar. I want to thank both of you for sharing such fantastic work. And I want to remind our viewers that we have a very deep website on the programs of the Cornell K. Liang Center for Wildlife Health. You can go to wildlife.cornell.edu. And I also want to thank Lisa Yang for all of her generosity that has helped make a lot of the work we're doing possible. So with that, we'll be back in the future with another One Health webinar, and I'm not quite sure what we're going to talk about yet, but we've got a whole line of faculty and postdocs and students who want to share information with you. So we'll be back. Thanks so much.